Welcome to this explainer. Today, we are looking at the biological equivalent of a massive industrial paradigm shift. We're talking about moving from painstakingly reading DNA one single letter at a time to processing billions of letters in a continuous cybernetic flow. Let's get right into it.
Now, to really grasp the scale of this revolution, you have to look at the legacy tech. For years, Sanger sequencing was the absolute gold standard. And hey, a classic 4-color chromatogram is a beautiful thing. But fundamentally, it's a massive bottleneck. It processes exactly one DNA read at a time through a capillary tube. Doing a whole genome this way literally took years and millions of dollars.
Next-generation sequencing, or NGS, completely shattered that limitation. Instead of just trying to read one sequence faster, NGS reads millions or even billions of them simultaneously. Think of it like upgrading from a single slow highway toll booth to a massive plaza with a billion lanes all open at the exact same time. Just look at this number, 600 billion. High-end modern NGS platforms can pump out up to 600 gigabases of data in a single run. That is 600 billion data points of genetic code, all generated in just a matter of days. It is a level of industrial-scale output that changed the lab forever.
First up is the reigning industry heavyweight Illumina, powered by their sequencing by synthesis method.
So, how do they actually pull off this massive scale? Well, DNA fragments attach to a glass flow cell, where they undergo bridge amplification to create localized clusters of identical molecules. Then the sequencing kicks off. Fluorescently labeled nucleotides wash over the cell, with each base glowing a totally different color. A high-res camera captures this synchronized light show, meticulously recording it one base at a time.
Now, the trade-off here is that the reads are relatively short, around 150 base pairs, but they are incredibly accurate, hitting 99.9% precision. When you combine that pinpoint accuracy with billions of simultaneous reads, it's pretty clear why Illumina became the undisputed leader for raw data output and cost efficiency.
Next on deck is the disruptor, ion torrent, with a wildly different physics-based approach called semiconductor sequencing.
What's really wild about ion torrent is what it completely leaves out of the machine. There are zero optics, no lasers, no cameras, no fluorescent dyes at all. Instead, it leverages the exact same manufacturing standards used to build the microchip in the smartphone you're probably holding right now. The sequencing chip literally is the machine.
Here's the magic. DNA templates sit in microscopic wells on the chip. Whenever a nucleotide base naturally attaches to a DNA strand, it releases a single hydrogen ion. The semiconductor instantly detects this tiny pH change as a microscopic voltage spike. It's basically a giant digital pH meter. One spike, that means an A was added. Two spikes, two A's. It's lightning fast and beautifully simple.
And of course, we have to talk about the pioneer that actually kicked off this entire revolution back in 2005, the 454 pyro sequencing system. 454 used emulsion PCR to amplify single DNA templates on tiny beads all suspended in oil and water. These beads got loaded into a picotiter plate. And to read the sequence, 454 relied on a cascade of enzymes, including luciferase. Yeah, that's the exact same enzyme that makes fireflies glow. Every single time a base attached, it released a tiny flash of light that was recorded on a pyrogram.
So if Illumina gave us volume and ion torrent gave us speed, 454 gave us length. It generated reads up to 1,000 base pairs long. Even though the platform is discontinued today, those pioneering long reads were absolutely crucial. They made the complex assembly of whole genomes possible during the early days of the NGS boom.
Let's put these three engineering marvels head-to-head and see how they actually stack up.
You can really clearly see the trade-offs right here. Illumina effortlessly wins on sheer throughput and the lowest cost per megabase. Ion torrent shines when you need rapid runtimes. We're talking just a couple of hours, because it bypasses all that optical hardware. And 454 historically held the crown for giving us the longest reads. Three highly distinct engineering solutions for three very different biological problems. But let's zoom out for a second. Why does all this raw industrial-scale data actually matter?
Because of NGS, we can map out the entire microbial diversity of environment, from literally just a handful of dirt. We can dive deep into clinical samples to pinpoint critical cancer biomarkers. We can sequence entire human genomes in a matter of days. NGS isn't just a tool, it's the fundamental engine driving modern biology and precision medicine.
Ultimately, these platforms forced a massive shift from a slow, linear process to a parallel flow of short, incredibly accurate reads. They essentially turned biology into an information science. Which leaves us with a pretty big question to think about. As sequencing a human genome becomes faster and cheaper than getting a standard MRI, how is society going to change when reading our foundational code becomes as routine as checking our blood pressure? Thanks for joining me on this explainer, and keep questioning the data.